we are heading towards the end of the chapter Principles of Inheritance and Variation. We have studied about genetic disorders and how they are brought about. In today's session, let us have a look at some of the most common genetic disorders in man. The first one that we have already seen is Down syndrome. It has individuals with short and broad hands, palmar crease, short stature, mental retardation, broader forehead, round mongoloid face and open mouth with a large tongue. It is because of the trisomy in the chromosome number 21. Next is the trisomy 13 or the Patau syndrome. It is also indicated by the chromosome formula 2n plus 1. It shows mental deficiency in children with deafness, minor cleft lip or cleft palate, polydactyly, and cardiac abnormalities. Next is the Edwards syndrome with the same chromosome formula and the chromosome nomenclature being 46 plus 18. These people are reported with multiple congenital deformities and low-set malformed ears. In Turner syndrome, as we have already seen, it has the chromosome type 2n-1. It is seen in females with retarded sexual development. They are sterile, have a short stature, skin webbing in the neck region, and cardiovascular abnormalities along with hearing impairment. Next is the Klinefelter syndrome, which has multiple chromosomal formulas. It indicates male fertility with a small testis, breast development, feminine voice, long lips, knock-knees, and talkativeness. In superfemales, that is the XXX syndrome, the chromosome formula is 2n plus 1 and they are usually normal genitally, but they have limited fertility and slight mental retardation. Let us now study some of these syndromes in detail. First is the criminal syndrome or the Jacob syndrome. This is found in males that are also known as the super males. Steinberg first described this condition in 1961 with chromosome number 2n equal to 46 and their chromosome formula being 44 plus XY. These XYY males are taller than normal males and they have speech and reading problems. There is a possible connection between the XYY karyotypes and the criminally aggressive behavior of these people. But this connection has not been fully established. Next is the poly-X syndrome, which is seen in females, and so these females are also known as the super females. Their chromosome number is 2n equal to 47 and their chromosome formula is 44a plus XXX. These females have no apparent physical abnormality. Their characteristics include strange facial features, webbing of skin, constriction of aorta, poor breast development, and underdeveloped rudimentary ovaries. It is seen due to XXX, that is one extra X chromosome, in chromosome number 23. Next we come to sex chromatin. Sex chromatin was discovered by a Canadian genetist named Murray L. Barr and E. G. Bertram in 1949. They were seen in the interphase nerve cell of female cats. This sex chromatin is therefore named Barr body after its discoverer, Murray Barr. They can be easily demonstrated in the cells of the buccal epithelium and their hair roots. Barr body is a highly condensed structure. It lies against the nuclear membrane of the interphase cells of these females. Human female has a positive sex chromatin and males have a negative sex chromatin. Right? Is it clear, everyone? Any doubts till now? This is the Barr body present inside the nucleus. A positive Barr body indicates female sex. Can we see very clearly presence of a Barr body inside the nucleus? According to a British genetist named Mary Lyon, one of the two chromosomes of a normal female becomes heterochromatic. This heterochromatic chromosome then turns into a Barr body. Thus, there is inactivation of one of the two X chromosomes in a normal female. And so, it is known as the dosage compensation or the Lyon's hypothesis. The number of Barr bodies is less than the number of X chromosome, or its number is nx minus 1. So it is 1 less than the number of X chromosomes. In Turner syndrome, there is sex chromatin negative, and in Klinefelter syndrome, the sex chromatin is positive. The Y chromatin or the Y body can also be identified as a bright spot by staining the cells with acridine dyes and viewing them under an ultraviolet microscope. The number of Y bodies also corresponds to the number of Y chromosomes. Clear, everyone? Any doubts? Now let us understand the process of inheritance related to sex. The sex chromosomes that are X and Y are unequal in size and shape. They are also heteromorphic and have homologous and differential regions. The genes that are located exclusively on the X chromosomes are called the X-linked genes, and the genes that occur on Y chromosomes are called the Y-linked genes or the Holandric genes, where holos means whole and andros means a man. So the genes present on the homologous regions in the sex chromosomes are called XY linked genes. We can see Holandric genes in a person with the most common feature being excessive hair on ears. Next syndrome is the G6PD deficiency, the most common feature, the excessive-linked recessive trait on ears, which is present. Next syndrome on X the G6PD deficiency is homologous in females. The sex-linked recessive single in males, and it causes the deficiency of on X chromosome 6-phosphate is homologous in female hydrogenase. Glucose 6-phosphate and it causes the deficiency of carrying out the hexose monophosphate dehydrogenase. This hexose glucose 6-phosphate shunt is required to produce for carrying out reduced glutathione monophosphate for protecting the sulfhydryl group. This hexose monophosphate and shunt is required to membrane during the reduced glutathione for protecting the sulfhydryl group of G6PD hemoglobin and erythrocyte membrane and during the result stress erythrocyte membrane ruptures. In absence of G6PD hemoglobin crystallizes. So G6PD deficiency as a result X-linked erythrocyte membrane which is characterized by red blood cell hemolysis after the exposure to oxidative drugs. In this, in most of the cases, the red blood cells are replaced, and so the oxidative drugs should be highly avoided. Next are some of the autosomal abnormalities. There is a large-scale possibility of autosomal aneuploidy in human beings, and the first syndrome we will study is the Edwards syndrome. It is due to trisomy of the 18th chromosome. It has malformed ears, receding chin, and defective nervous system in individuals. Next is the Patau's syndrome. It is due to trisomy of 13th chromosome. The individuals show hair lip and cleft palate. We can see different symptoms of Edwards syndrome like a small mouth, small jaw and short neck, shielded chest with a wide sternum and widely set nipples. The back part of the skull is extremely prominent. These individuals are dysplastic and have malformed ears. They have clenched hands with overlapping fingers and flexed big toe with prominent heels. It is due to trisomy of chromosome number 18. Next is the Patau's syndrome. Such individuals develop a small head and absent eyebrows. They have cleft lip or cleft palate with dysplastic or malformed ears, clenched hands and polydactyly, and undescended or abnormal testes. Some other autosomal disorders include polydactyly, where a person has more than 5 fingers, achondroplasia, which is an inherited dominant autosomal disorder, where the long bones do not grow properly, and so the individual is dwarf. Their intellect is not affected. Then it's muscular dystrophy, which is due to dominant autosomal gene, and it is of 4 types: myotonic, facio-scapulohumeral, aculo-pharyngeal, and scapulo-peroneal. There are different types of polydactyly, for example, preaxial or radial, the central, and the postaxial or the ulnar. Can you name one famous personality having preaxial or radial polydactyly? Yes, he is a film star. Very good. Hrithik Roshan has preaxial or radial polydactyly. Let us now come to some of the genetic diseases in man. There are many diseases in man due to gene mutations. A mutated person may become incapable to produce specific enzymes, and so there are inborn errors in its metabolism. First is the chondrodystrophic dwarfism. It is a dominant autosomal mutation, where most of the people are homozygous for recessive alleles. The presence of one dominant C results in premature closure of the growth areas of long bones of arms and legs. This results in shortened and bowed arms and legs. Next is the Huntington disease. It is caused by a dominant gene on chromosome number 4. The mutated gene causes abnormality by producing a substance. This substance interferes with normal metabolism in the brain and leads to progressive degeneration of the brain cells. After the onset of symptoms, within 10 to 15 years, the individual dies. Next is neurofibromatosis. It is also known as the von Recklinghausen disease, and it is caused by a gene mutation on chromosome number 17. The affected individuals may have 10 spots on the skin, which later increase in number and size. They are small benign tumors, and they are called the neurofibromas, and they occur on the skin on various organs. This is an example of neurofibromatosis. With axillary freckles, cafe-au-lait macules, and the buttonhole sign. We can also see the Huntington's disease, where a mutant Huntington protein affects the chromosome number 4 and cause degeneration of the brain cells. Next is the Tay-Sachs syndrome. It is a disease which results from lack of dominant gene on chromosome number 15. This gene is responsible for production of hexosaminidase, and so a subsequent storage of its substrate is not possible. A fatty substance known as glycosphingolipid is found in lysosome. Is the G6PD deficiency. These patients suffer from defect of vision, the sex-linked recessive trait, which is present muscular weakness, loss of mental chromosome and physical control, and death. Is homologous in female. You can see the fatty substance getting deposited in the brain. Single in male, thus the nerve cells in the brain that causes the death get deficiency, and the neurons in the brain and the spinal cord are degenerated because of the deposition of this fatty substance. Glucose 6-phosphate dehydrogenase. Next is Alzheimer's. But Alzheimer's disease is named after a German neurologist named Alzheimer. This is a degenerative brain disease. It is characterized by severe memory loss, confusion, restlessness, phosphate, speech disturbance, erosion of personality, judgment, an inability to perform the reduced glutathione. For protecting, it is a form of dementia, and the sulfur it can occur groups of hemoglobin normal the erythrocyte membrane contains 5% during the karyotypically normal individuals. Oxygen over the age of 25 develop Alzheimer's, and 25% of people over the age of 80. An absence of G6PD hemoglobin are caused by this disease. The brain of Alzheimer's patients, as a result, a remarkable loss membrane rupture of neurons. So, G6PD deficiency is an X-linked recessive disorder which is characterized by red blood cell hemolysis. a normal person, amyloid B protein is produced. This protein is produced from a large number of amyloid precursor proteins. In people with Alzheimer's disease and Down syndrome, there is a gene on chromosome number 21. Genetic mapping has demonstrated that the gene for amyloid beta protein is also located in chromosome number 21, which encodes an amyloid precursor protein. This is enzymatically cleaved in order to produce the amyloid beta protein. In Alzheimer's disease, there is a zinc buildup in the brain along with deposition of copper. As we can see in the image, the brain changes in Alzheimer's disease. This is the normal cortex, while the cortex of Alzheimer's patients shrinks in their size. Their ventricles are enlarged, and the hippocampus is also shrinked. These are the normal brain nerve cells, whereas these are the damaged nerve cells with tangles and amyloid plaques in Alzheimer's patients. So we can clearly demarcate the difference between a healthy brain and an Alzheimer's brain. Next disease is cystic fibrosis, which is a lethal genetic disease due to a recessive mutation on chromosome number 7. Here the body produces an abnormal amount of glycoprotein. This glycoprotein interferes with the salt metabolism in the body. Thus, the mucus is secreted by the body, which is abnormally viscid and blocks the passage in the lungs, liver, and pancreas. Next is the Marfan syndrome, which is due to dominant mutation resulting in the production of abnormal form the long bones of the body grow longer in this disease, and the fingers are known as the spider fingers or arachnodactyly because of their increased length. Next is thalassemia. Thalassemia is nothing but human anemia, which develops due to an autosomal mutant gene. When this gene is present in a double dose, the disease is severe thalassemia major, resulting in death of the individual in its childhood. Heterozygous people have milder disease known as thalassemia minor. It is also known as the Cooley's anemia. Amyloid B protein is produced. This protein is produced from a large number of amyloid precursor proteins. In people with Alzheimer's disease and Down syndrome, there is a gene on chromosome number 21. Genetic mapping has demonstrated that the gene for amyloid beta protein is also located in chromosome number 21, which encodes an amyloid precursor protein. This is enzymatically cleaved in order to produce the amyloid beta protein. In Alzheimer's disease, there is a zinc buildup in the brain along with deposition of copper. As we can see in the image, the brain changes in Alzheimer's disease. This is the normal cortex. Their ventricles are enlarged and the hippocampus is also shrinked. Amyloid plaques in Alzheimer patients. So we can clearly demarcate the difference between a healthy brain and an Alzheimer's brain. Next disease is cystic fibrosis, which is a lethal genetic disease due to a recessive mutation on chromosome number 7. Here the glycoprotein interferes with the salt metabolism in the body. Thus, the mucus is secreted by the body, which is abnormally viscid and blocks the passage in the lungs, liver, and pancreas. This is due to dominant mutation resulting in the production of abnormal The long bones of the body grow longer in this disease, and the fingers are known as the spider fingers or arachnodactyly because of their increased length. Next is thalassemia. Thalassemia is nothing but human anemia, which develops due to an autosomal cause. The disease is severe thalassemia major, resulting in death of the individual in its childhood. Heterozygous people have milder disease known as thalassemia minor. It is also known as the Cooley's anemia. These people are unable to produce the beta chains. Their hemoglobin is like that of fetus, which is unable to carry out normal oxygen transporting function. And individuals with formed red blood cells. Next is the Gaucher's disease. It is also a genetic disease which is associated with abnormal fat metabolism and is caused by the absence of enzyme glucocerebrosidase. Enzyme glucocerebrosidase is responsible for proper processing of lipids in our body. Non-processing of lipids results in accumulation of fatty material in spleen, liver, bone marrow, and brain. Thus, these organs swell and the patients die by the age of 15 years. Some of the clinical symptoms of Gaucher's disease include pulmonary involvement, hepatosplenomegaly, skeletal involvement, progressive neurologic symptoms, thrombocytopenia and anemia, and so on. Next is galactosemia. It is also an inherited autosomal recessive trait, and the affected person is not able to convert galactose to glucose. Galactosemia is due to the deficiency of the enzyme galactose phosphate uridyl transferase. Milk is toxic to galactosemic infants, and so these children die at the age of 3 years. Next is the taste blindness. It is also a genetic trait. It is because of PTC, which is phenyl thiocarbamide. It is a compound of nitrogen, carbon, and sulfur with a sour taste. About 30% people lack the ability to taste PTC, and it is also transmitted by a dominant T gene. Their D genotypes are capital T, and capital T, small t. Now, what are human hereditary traits? There are about 200 traits in human beings that are transmitted from one generation to another. Thus, the inheritance in man conforms the genetic principles that govern inheritance in other plants and animals also. These traits include hair, skin color, eyes, eyelashes, vision, lips, nose, nostrils, hearing and speech, stature, blood clotting, blood groups, Rh factor, ear, cheeks, and tongue rolling. Different methods are approached for their studies. These include pedigree record of families, population genetics to analyze the fate of characters in populations, human cell culture to know the biochemical basis of inheritance of many characters, human cytology and study of twins. Human chromosomes have many numbers and different karyotypes. The diploid chromosome number in human beings is 46. Your each cell has 23 pairs of chromosomes, out of which 22 are autosomes and 1 is a pair of sex chromosome. The sex chromosome controls the sex of an individual. Karyotype is systematic representation of the chromosome complement of a cell or an organ. It shows the number, size, type, form and position of the centromere of the chromosome. So, here we finish off with the chapter Principles of Inheritance and Variation. We have studied a lot of concepts of Mendel, his studies, genetic disorders in much detail in this chapter. All the topics that are discussed in these sessions are very important from your examination point of view. It also leads to a clear understanding and forms a basis